Good morning, respected faculties. Myself Kratu A Patel from MSc Analytical Chemistry, St. Xavier's College, Ahmedabad. Today, I am going to discuss in detail about gas chromatography. So, as we know that a technique used for the separation of a mixture by passing it in a solution or suspending it through a medium in which the components move at the different rates is termed as a chromatography. In contrast to other chemical methods of separation, this analytical technique is very quick. And in chromatography, the components of a mixture are distributed between two phases, the one which is a stationary phase having a large surface area, and the other is a mobile phase which moves continuously in contact with the stationary phase. The stationary phase, such as porous solid, and mobile phase as a gas or a liquid is used. The basic different types of chromatography techniques are liquid chromatography, gas chromatography, thin layer chromatography, and paper chromatography. Furthermore, gas chromatography is subdivided into gas solid chromatography, gas liquid chromatography, gas bonded phase chromatography, which is relatively a new technique. This chromatography technique was developed by James and Martin in the year 1952. Here, the sample taken first is vaporized and separation occurs in a gaseous form. The column is heated and maintained at temperature around 350 degrees Celsius to 400 degrees Celsius in an oven. And generally, the liquid samples have maximum boiling point 250 degrees Celsius can be separated by gas chromatography. Further, they have to be thermally stable, and in spite of this limitation, the technique is used to a large extent in each segment of industry. Now moving towards the principle of gas chromatography is that a sample is vaporized by injecting into a heated system and eluted through a column by inert gaseous mobile phase, and then it gets detected. There are approx 2 lakh GC instruments used in the worldwide, and the primary components of a gas chromatography are carrier gas system, sample introduction system, column, which is relatively a versatile component, then GC detectors and recorders. The labeled instrumentation diagram of gas chromatography is shown here. The first component of a gas chromatography system is the carrier gas system. A flow rate of a carrier gas is an important parameter on which the efficiency of a separation depends. A flow meter is used to measure the flow rate of a carrier gas. For example, soap bubble flow meter, which is simple and most inexpensive one. Carrier gas is used as a mobile phase in a gas chromatography system, and it must be chemically inert. And the common used gas include nitrogen, helium, argon, and carbon dioxide. The choice of a carrier gas is often dependent upon the type of detector which we use. A carrier gas also contains a molecular sieve which is used to remove water and other impurities. Gas clean filters are used before the instrument inlet, which reduces the level of impurities and thus it improves our trace analysis. Autosamplers are also used in the gas chromatography system, which automatically takes the sample and insert it in the system for our analysis. The sample used must be of a suitable size and introduced instantaneously as a plug of a vapor. A slow injection or an oversize causes peak broadening and poor resolution. Therefore, a long needle syringe are used to prevent band broadening due to diffusion of a sample band. Also, electrical heaters are used which controls the temperature electronically, and the temperature is maintained at 10 to 20 degrees Celsius higher than the boiling point of a component so that it does not vaporizes in the column. As we know that light gases are likely to be diffused through the needle of the syringe, and hence to introduce the gaseous samples precisely, the gas sampling valves are used. For quantitative analysis, sample splitters are also used, which splits the samples into two parts, and the smaller part is introduced in the column and the bigger part is vented off. And the gas filled in the loop is carried to the column along with the carrier gas. A microscopic layer of liquid or a polymer on an inert solid support inside a piece of a glass or a metal tubing is known as a column. It is considered as the heart of a gas chromatography system, and here the column is used as a stationary phase. It is a tube-like structure where the separation is performed in the chromatography technique. It is made up of stainless steel or a glass. The diameter ranges from 15 to 30 meter long. The diagram view of chromatographic column is shown. Generally, there are two types of columns, packed column and capillary column. Packed column contain a finely divided inert solid support material such as diatomaceous earth, coated with liquid stationary phase. Mostly packed columns are 1.5 to 10 meter in length, having an internal diameter of 2 to 4 millimeter. Capillary columns are also known as open tubular columns, having an internal diameter of few tenths of a millimeter. The basic types of capillary columns are wall-coated open tubular, support-coated open tubular, polar porous layer tubular, few silica open tubular, out of which first and second one are mostly used in the labs as a chromatographic columns. This is the image of packed and a capillary column. The stationary phase filled inside the column can be either solid or a liquid. If the stationary phase is a solid, then it is said to be gas solid chromatography, which is abbreviated as GSC. And if the stationary phase is a liquid, then it is said to be gas liquid chromatography, which is abbreviated as GLC. GLC is used in most of the labs because it is based on the partition chromatography technique, and the distribution coefficients of GLC are much smaller compared to GSC. Also, the retention time is comparatively shorter in GLC. The activity occurring in the column is the sample coming with the mobile phase is in a gaseous form and which passes through the column. The molecules which are having lower boiling point will move faster in the column, and the molecules coming out of the column are known as elutes. The first elute coming out will be detected by the detector and send the signal to the computer. In case of higher boiling point molecules, as they cannot move faster, the temperature of oven is adjusted or changed in running chromatography process, and such process is known as temperature programming. If the boiling point of both the components is same, then we consider the size of individual components. The smaller molecules move faster and the larger molecules will move slower. Talking about the detectors of the gas chromatography, the detector is used to detect and quantify the different components of the sample as they emerge out from the column. The requirements of an ideal detector are: it should be highly sensitive, physically suitable, capable to operate up to maximum column temperature, easy to operate, response to the compounds of which analysis is required, no response to undesirable compounds, it should be non-destructive to the samples in case of preparative work, and it should be inexpensive. The different types of detectors used in gas chromatography are flame isolation detector. It is selective for most organic compounds. Hydrogen gas is used as a carrier gas. It can detect up to 100 picograms. Its dynamic range is 10:7. The second detector is thermal conductivity detector. It is a universal detector which can detect all the types of compounds, and the carrier gas usually used as hydrogen, helium or any reference gases. It can detect up to 1 nanogram. Its dynamic range is 10:7. The third type of detector is electron capture detector. It is selective for halides, nitrates and peroxides. We need to make up the gas as a carrier gas. It can detect up to 50 femtograms, having dynamic range up to 10:5. The next detector is the thermionic detector. It is similar to flame ionization detector. It is selective for nitrogen and phosphorus compounds. It is 500 times more sensitive than flame ionization detector for the phosphorus and 50 times more sensitive than flame ionization detector for nitrogen, and it is bad for carbon compounds. Fifth type of detector is nitrogen phosphorus detector, and the name itself gives an idea that it is selective for nitrogen and phosphorus compounds, and the hydrogen gas is used as a carrier gas. It is a type of mass flow and it can detect up to 10 picograms, having a dynamic range of 10:6. The sixth type of detector is flame photometric detector. It is selective for phosphorus and sulfur compounds, and here the support gas, which is a carrier gas, used as a hydrogen gas, and it uses optical filters to isolate the signal. The next detector is the photoionization detector. Here the column is irradiated with intense UV light source which ionizes the molecules, and it measures the ions with electrodes in detector cell. The next is the GCMS chromatography. When the mass spectrometry is used as a detector, we can say that the chromatography is said to be GCMS chromatography technique. It is highly sensitive. It can be very selective in selected ion monitoring (SIM) mode. It is powerful for analyzing qualitatively and quantitatively. Another kind of mass spectroscopy, such as ion cyclotron MS, which gives higher resolution. The signal from a gas chromatograph is continuously recorded as a function of time. Generally, with the help of potentiometric strip chart, it gets recorded. Mostly a recorder records 1 to 10 millivolt with the full scale deflection of 25 cm and a response time of 1 second or less than it. A recorder have a facility through which the smaller peaks can be enlarged and bigger peaks can be suppressed. A recorder can speed up the sample analyzation between 5 to 50 mm per minute. A recorder traces a peak and the area under the peak is directly proportional to the concentration of solute, and there are no such methods used to calculate such area, so electrical integrators are used. Time between the sample injection and the analyte peak reaching a detector at the end of a column is termed as retention time TR. The limitations of gas chromatography are: it is limited to volatile samples, it is not suitable for thermally labile samples, samples should be soluble and do not react with the column. During injection of gaseous sample, proper attention is required. Advantages of gas chromatography are: it improves the resolution using different parameters such as temperature programming, gas flow rate, split ratios. It also improves the analysis speed in couple of minutes than thin layer chromatography. It prefers wider sample choice from liquid gases and dissolved solid. It is more sensitive with the help of the different types of detectors, and due to column choice, the different types of separations can be performed, and it is non-destructive while testing. The uses of gas chromatography are: it is used to detect blood or alcohol level in patient's bloodstream. It is also used to compare a sample found at a crime scene to sample from the suspects. It is also used to determine the level of pollutants in the water supply. It is also used to purify a chemical needed to make a product. So that's all for the gas chromatography technique. Thank you.